Hello everyone, welcome to another unboxing of lab reagent videos. This is Dr. Bonik from the lab 7. So today we're going to look in the Arba Quick Malaria Kit. So this kit is for in vitro diagnostic use only. So first, when we purchase any such kits, we have to see what's written here. So as we can see that this is the Arba Quick Malaria Antigen Plasmodium falciparum or Plasmodium vivax test card. So first what we have to do is we have to tally the lot number with the bill that we have received and check the manufacturing date and the date of expiry. Next, if we come here, we can see that this box contains the test cards in pouch. There are 25 pieces in this box. There is also supposed to be a buffer solution, a sample dropper and instructions for use. Another thing to note is that on the outer surface of each such box, we see the temperature. So here the temperature is from 1 to 40 degrees Celsius. So this kit can be kept at room temperature, but it is advised that after each day's work, we keep it in the refrigerator, but not in the freezer. So now let's see what's inside the kit. So before opening the kit, it is advisable that we write the date of opening of each kit and if we want the date of expiry. So let's have a look inside. So when we open this kit, what we can see is that these are the card, the rapid test, and these are the cards, the malaria antigen test card. The principle is the immunochromatography, and here we also tally the lot number, the date of manufacturing and the date of expiry. 25 such cards have been provided. With this, we also have the plastic droppers for the sample. We have thank you for choosing TransAsia, and we have the buffer solution. So here also in the buffer solution, we check the lot number, the date of manufacturing, the expiry date, and here also we can see the temperature has been mentioned. Now moving on, we also have a customer feedback form which if we want we can submit, especially in case of any complaints. The most important thing before using any kit is the kit literature. So here also TransAsia has provided the one-step malaria antigen rapid test kit. So first, in any such kit, what we have is the intended use, the introduction, the principle, the kit components, storage and stability instructions, how to collect specimen, and the procedure along with the limitations and inferences. So first, this is a malaria antigen Plasmodium falciparum and Plasmodium vivax test. Now what is the antigen? So we must know that for Plasmodium falciparum, the antigen is histidine-rich protein 2, which is specific for Plasmodium falciparum, while for Plasmodium vivax, it is lactate dehydrogenase. Since there are two antigens, this is known as malaria parasite dual antigen test. So the principle of this test is that it targets these antigens and we look for a red line. Okay. Now we will go into this later again in details. Now specimen collection, EDTA blood is usually used and we have to use it and do the test more or less within 15 to 30 minutes because we have to wait for 30 minutes and read the test. So what is the procedure? So first we have to allow the kit components to come to room temperature. We have to remove the foil from this kit, place it on a flat dry surface. With this dropper, we take a 5 microliter disposable dropper and we draw the sample blood and put it in the vial. So this is the kit and it is the, if we open it, we get a test cassette. Okay. So in this test cassette, we see that there are 3 lines. So first line is control. Then we have Plasmodium falciparum, Plasmodium vivax. S is the sample vial and B is the buffer vial. So first with the dropper, we give the sample in the sample vial and then we give the buffer in the buffer vial. Next what happens? We wait. Whatever might be the case, even if it is positive or it is negative, there has to be one red line in the C, that is the control zone. Okay. And next, if it is Plasmodium vivax or Plasmodium falciparum, we will have one line in either Plasmodium vivax or in either Plasmodium falciparum. But we must note that the C line has to be positive to interpret the test. If by any chance the C line is absent, then the test is invalid. Other things to note while doing this test is that the known relevant interfering factors is that hemolytic samples, rheumatoid factor containing samples, lipemic and icteric samples can lead to impairment of test results. So that is all. Thank you. See you in another video.